Alright, this is the Chapter 8 video. We're going to be covering microbial metabolism. This is a two-part video series where the first part we're going to talk about enzymes, enzyme function, why enzymes are important, and then the second video will be over glucose metabolism and how cells generate ATP and the different ways bacteria are able to do that. Make sure that you complete the study guide for Chapter 8 found in the course pack. Pause the video, take your notes, and then when you're done with the video, you can complete the online e-learning quiz. So metabolism is the sum of all the chemical reactions within a living cell. These reactions can be either catabolic, which are energy harvesting processes in which the bacteria produce ATP. That ATP is stored in the cell and used during anabolic reactions, which are building processes. So catabolic is a breakdown, anabolic is building. And there's thousands of different reactions that occur in a cell that control different mechanisms at any given time. These reactions occur in what are referred to as metabolic pathways, or I will often call them biochemical pathways. Metabolic pathways are just a series of chemical reactions required to break down or build a cellular component. So there's a substrate, which is basically like the food source. There are intermediates, which is when the molecule is slightly changed. And then there are products or byproducts, which is the end product overall. And with a metabolic pathway, where we have this arrow here, an enzyme would be involved in creating the intermediate from the starting compound. The enzyme's role in the metabolic pathway is that they help facilitate the pathway. They are made of protein, and they're referred to as biological catalysts. And what a biological catalyst does is it lowers the activation energy of a chemical reaction. If it lowers the activation energy of the chemical reaction, the reaction occurs faster. So activation energy is the amount of energy needed to start a chemical reaction, which is what the enzyme role does, is it lowers the amount of energy. The reaction can be, this reaction can kind of be explained with this figure here, although there is one problem with this. Imagine yourself walking up a hill carrying a big boulder. You're going to use a lot of energy to do that. Okay, so the relative amount of energy over here on the left over time, it's going to take you a while to do this. It's going to require a lot of energy. Once you get to the top of the hill, you start the reaction by dropping that boulder, and you create the end products by smashing that boulder into little bits and pieces. Okay, with an enzyme, you lower the amount of energy needed to create the same overall end product. The problem with this image is that, like I said, the time would actually be reduced as well as the amount of energy. So there is that problem with this figure. This slope here should be much further over here to the left. The image in the book kind of demonstrates this, although the illustration of the human and the wheelbarrow are not very artistic. We name enzymes usually by adding ASE at the end of the substrate. So just a couple examples: lipases work on lipids and proteases work on proteins. And for the most part, enzymes are substrate specific. So a lipase will only work on lipids and a protease will only work on proteins. And we can explain this enzyme specificity in what we call a lock and key theory, or also referred to as the induced fit model. Here we have a substrate, here is our enzyme, and with the enzyme there's the active site, and the enzyme is representing the lock here, and the key, you can see, fits exactly inside of that active site. So we're all familiar with keys. We have one that starts our car, opens our house, things like that. Those keys are specific for that lock. I'm going to pause the video here for a second. I'm going to go to a writing board and do another demonstration. All right, so when I draw an enzyme, I typically draw Pac-Man. Everybody's familiar with Pac-Man. should be. And Pac-Man in my class, he eats pizza. So Pac-Man's the enzyme. And like I said, that's the lock. The active site is Pac-Man's mouth. Pac-Man eats pizza, that's the substrate. Or the key. The substrate fits right into Pac-Man's mouth. All right, so Pac-Man eats pizza. And our completed enzyme here with the substrate is referred to as the enzyme substrate complex. I apologize for the quality of my handwriting here. Once Pac-Man has the substrate in his mouth, Pac-Man will chomp down basically on the substrate and create end products. So here we'll just draw 2 slices of pizza then, indicating that he did some work. So this would be a breakdown of this one substrate. So this would be referred to as a catabolic reaction. Catabolic, again, breaking down. Some enzymes are complete on their own just as the protein component. Others have non-protein components. Those non-protein components are referred to as either a cofactor or a coenzyme, which is the non-protein component. So we call these enzymes that require a non-protein component apoenzymes. Okay, so they're basically an incomplete protein. The cofactors are either inorganic ions, such as iron, magnesium, zinc, and coenzymes are derived from vitamins, and they typically can't be synthesized by certain organisms. Therefore, we have to consume vitamins in order to help complete the active site. So that's what the cofactor does, is it helps complete the active site. Here we have this blue blob indicating the apoenzyme, which is the protein component, and the cofactor fits into basically the active site portion, and then helps form the actual active site. Going back to the drawing board here, I usually draw Pac-Man, my apoenzyme, a little spot here. So this is an apoenzyme, and we'll still require him to eat the substrate pizza, but here we need that non-protein component. So that would go right into this spot here, completing the active site. Okay. Typically with enzymes and substrates, there's an attraction and affinity by some type of electrochemical charge. So some positive charges here, and that the cofactor would actually provide a crucial charge so that there is that attraction. So there might be some negatives here, opposites attract. Now that this enzyme is complete with the cofactor, oops, I'm supposed to say cofactor. Now the substrate can actually enter into Pac-Man's mouth and then form that enzyme substrate complex. Okay, and again, it's the electrochemical charges associated with the substrate and the opposite charges with the enzyme that help that attraction occur. Now there are different factors that influence enzyme activity. So the cell's ability to survive in extreme temperatures or pH, if we remember back from chapter 5 with the alkalophiles and the acidophiles, or the psychrophiles and the thermophiles, it all has to deal with their enzyme's ability to actually resist those extreme temperatures and pHs. Okay, so things that can influence the activity would be things like temperature. So if temperature increases too much, the proteins can denature and lose function. So usually as temperature goes up, there will be some optimal range, just like when we generated the growth curves in Chapter 5 lab. And then you saw that sharp decline in growth and eventually going down to nothing because those proteins denature. Denaturation of a protein means that the folding, the way that those proteins are folded, become unraveled, or they either bind even tighter, no longer making that protein active. Again, pH can influence the activity. Like we said before, organisms have optimal pH ranges in which they can function. Also, substrate concentration influences enzyme activity. Basically, if you increase the substrate, the enzyme food, more and more, that enzyme activity will level off. So the enzyme can only work so fast. I like to kind of use an analogy. If you're an enzyme and your substrate is pizza, and you're in a room and somebody's delivering pizza to you, you can only eat that pizza at a certain rate. Your activity is going to level off. It doesn't matter how much pizza is in that room, you can only eat it at a certain rate. Without enzymes, the energy yielding reactions could occur, but the rates would be extremely slow. Basically, life as we know it would not exist. Now, there's apoenzymes, there's regular enzymes, but then there's also enzymes called allosteric enzymes. And allosteric enzymes are used to regulate or control the activities of different key enzymes. So they have what's called an allosteric site that's separate from the active site. So molecules can bind to this and distort the enzyme's active site, which then again prevents them from working. Here this image has an enzyme, the active site, allosteric site, and the allosteric inhibitor can bind to this and changes the shape. Alright, so here I have drawn an allosteric enzyme, the protein component, the enzyme Pac-Man, allosteric site, the site separate from the active site here. And here we have this inhibitor molecule that can bind to this. And once this inhibitor binds to this, like we said, it distorts the active site. So here Pac-Man's going to grow some teeth. He's got some big chompers now, where this pizza is not going to fit into Pac-Man's mouth. So the allosteric enzymes have these sites that allows the cells to regulate different pathways. I'll give you an example here in a moment. But if they have these sites, they can no longer, the biochemical pathway will no longer be functioning. So cells apply these allosteric enzymes in what's referred to as feedback inhibition. So what happens is the end product of a pathway can act as an allosteric inhibitor of the first enzyme in the pathway. So the end product shuts down the pathway. This allows the cell to control the synthesis or building of certain amino acids. An example here is E. coli in the presence of the amino acid isoleucine. Isoleucine actually allosterically inhibits the first enzyme in the pathway that would actually synthesize isoleucine. So the shutdown, or the pathway gets shut down. All right, so here I have a little bit of a pathway here to demonstrate this feedback inhibition. This would be our first enzyme in a pathway. This is our first substrate. This is the enzyme substrate complex, and then here is what we would call an intermediate. Now this intermediate is actually the substrate for enzyme 2 in the pathway. So this intermediate goes into Miss Pac-Man's mouth here, and she would chomp on it and create a product. Now as long as there's substrate, there's going to be intermediate. As long as there's intermediate, there's going to be product. So as product builds up, as this pathway resumes, when this concentration of the product gets high enough, it feeds back and attaches to the allosteric site of that first enzyme in the pathway. Once the allosteric site has the inhibitor in that site, again, Pac-Man grows teeth, and it will actually shut down this entire pathway. So then we don't have this enzyme substrate complex, we don't have intermediate, and then eventually, basically, we don't have product. Okay, so cells will do this if they're synthesizing amino acids. So here, with that example of isoleucine, if this product is isoleucine, the cell can only use that amino acid so fast to make the actual proteins. So having this feedback process, the cell basically conserves energy by not producing intermediate and by not producing product. Now, as this product goes away, the cell utilizes, it makes proteins. Well, this process is reversible, and the pathway can resume. Oops. So Pac-Man would lose his teeth if that allosteric inhibitor or the end product dissociates. So it's a concentration-based inhibition. So concentration of the product goes up, that product can bind and shut down the pathway. As it depletes, this can disassociate and resume that pathway. So one of my favorite questions to ask usually is why do we care about enzymes? Just like I like to ask, why do we care about cell wall structure? Well, we care because we can design antibiotics that interfere with metabolic pathways of bacteria. So there's a group of antibiotics called antimetabolics that do this. We also care because biochemical testing is done in order to test for an enzyme, bacteria's ability to produce an enzyme. If they produce the enzyme, they will produce a product, and that's what we usually test for. So with enzyme inhibition, like I said, we can design antibiotics that can interfere with a pathway. So there's two different types. We have competitive inhibitors, we have non-competitive inhibitors. Competitive inhibitors, they compete for the active site. So they compete with the substrate for the active site. And they can do this because they have a chemical structure that's similar to the substrate. So here we have our competitive inhibitor. Here's our substrate. See, the square piece would fit right into that enzyme. Here, if we have the competitive inhibitor in a high enough concentration, we can bind that to there and block the actual substrate from binding. So the example would be sulfonamides, sulfa drugs. What it does is it competes for the active site on bacterial enzymes that convert this chemical called PABA into folic acid. It's not really important what PABA is, but it gets converted to folic acid. And sulfa has a similar chemical structure to PABA. So it prevents that substrate PABA from binding, therefore we don't get folic acid produced. The bacteria doesn't. Folic acid is required for DNA and RNA synthesis, so if you have no folic acid, you have no DNA and RNA synthesis, you have no cell replication. So you can actually stop the bacteria from growing by adding sulfa. Now, with antibiotics, we want them to be selectively toxic. So that means it isn't going to affect the human since we cannot synthesize folic acid. So we consume folic acid in our diet. Of course, you can have allergic reactions to antibiotics such as sulfa, but in general, it's not going to interfere with our folic acid pathways because we don't have one. Now, non-competitive inhibitors, what these do is they attach to allosteric sites. So here's our non-competitive inhibitor and an allosteric site. Well, again, if you attach an inhibitor to an allosteric site, it's going to distort that active site so the substrate can't attach. Now, there aren't any antibiotics that work in this fashion, but it may explain the toxic effects of certain metals like arsenic and lead, how they affect not only us as humans, but bacterial growth as well. Like I mentioned, enzymes are important for biochemical testing as well. Since every bacterial species produce different enzymes, this allows them to produce different products. It gives them different metabolic byproducts. Here's a slide showing you Streptococcus pyogenes, and here's Staphylococcus aureus. Now, gram staining and microscopic visualization alone is not enough because these organisms can actually look very similar to one another. Streptococcus means chains of spheres, but here you actually see clumps in there as well. Staphylo means clumps, spheres. Here you see, well, you see some chains and you actually see clumps as well. So microscopic evaluation is not enough for us to identify unknown bacteria. So an example that we can do to differentiate between Staphylococcus and Streptococcus, biochemical test, catalase test. I think we mentioned this before in chapter 5. Catalase is produced by aerobic respiring organisms to detoxify hydrogen peroxide. So this is a simple test to differentiate between Staph and Strep. Streptococcus are always catalase negative. They're considered obligate fermenters, meaning that they can survive in the presence of oxygen, but they do not use oxygen. So those were the aerotolerant or the fermenters from chapter 5. Staphylococcus are catalase positive. So to do this test, you basically place a few drops of hydrogen peroxide on bacterial growth, and you look for the presence of bubbles. Here, Streptococcus, no bubbles. Staphylococcus, bubbles. So Staphylococcus is catalase positive. So again, this is some basics of metabolism and why enzymes are important. It's very important to know, be familiar with the enzyme anatomy, with the active site versus the allosteric site, what happens when an allosteric site has an inhibitor bound to it, what feedback inhibition means, and things like that. So this will conclude the first video, and I will get to creating the second one.